Dr. Ali Nurmansyah, the head of the Department of Plant Protection, IPB University. To Pak Ali, the time is yours.
Okay, thank you, Bulia. That gives time for me to give welcome speech to the seminar. Assalamualaikum warahmatullahi wabarakatuh. Good morning, everyone. Good morning, everyone. First of all, I would like to extend my deepest thanks and appreciation to our keynote speaker, the Honorable Associate Professor Dr. Salmah Yaakop from Biological Sciences and Biotechnology, Universiti Kebangsaan Malaysia, and two of our speakers, the Honorable Dr. Takumasa Kondo from Agrosavia, Palmira Research Station, Colombia, and Dr. Purnama Hidayat from Department of Plant Protection, Faculty of Agriculture, IPB University, who will deliver their presentation to all of us. Also, I would like to say a big thank to you all, the audience, who attend this seminar. This seminar is the first seminar in a series webinar that we will hold in this year. This webinar carries the theme Exploring the Nexus of Science and Digital Technology, Contemporary Insight into Insect Systematic and Biodiversity. I think this theme is very important and very appropriate to current technological development nowadays, where digital technology has entered all aspects of human life, from personal needs to public needs. The field of insect systematic and biodiversity also really needs the advance of in digital technology so as not to be left behind. And I'm sure that the material that will be presented by speaker in this webinar will be able to provide new insight and thinking in the field of plant protection, especially to our student in the Department of Plant Protection. And finally, I would like to say thank you to all persons that already prepare and also manage this seminar, so this webinar can go smoothly. And also, I would like to say thank you to all of the organizations that support this webinar in today. Those are my brief remarks, and welcome and enjoy this seminar. Assalamualaikum warahmatullahi wabarakatuh.
Okay, thank you, Pak Ali, for your opening speech. Before we begin, I would like to take a few moments to explain the rules for today's seminar. First, please ensure your microphone is switched off or mute so we don't disturb the speakers and other attendees. We encourage you to participate and ask questions about the seminar topic during discussion or Q&A session. You can raise your hand by clicking raising hand icon or send the question through the chat room. Please keep your question brief and to the point to allow for a productive discussion. Please ensure you stay for the seminar until it finishes at 11 a.m. as it will provide a holistic understanding of the topic. So the topic of today's seminar is exploring the nexus of science and digital technology, contemporary insight into insect systematics and biodiversity. We have exciting speakers lined up for you today. We hope you will find their presentation and discussion informative, valuable, and engaging. There are 3 speakers who will give the presentation in this webinar. Firstly, our keynote speaker, Associate Professor Dr. Salmah Yaakop from Biological Sciences and Biotechnology, Universiti Kebangsaan, Malaysia. Next, we have Dr. Purnama Hidayat from Department of Plant Protection, IPB University. And last but not least, we have Takumasa Kondo, PhD from Agrosavia, Palmira Research Station, Colombia. The seminar will be conducted by moderator Dr. Idam Sakti Harahap from IPB University. So I will read the CV of Pak Idam first. Bapak Dr. Idam Sakti Harahap is from the Department of Plant Protection, IPB University, with the expertise in entomology, especially in urban pests. He was finished his bachelor and master degree in IPB University and his PhD in Clemson University. You can read in the screen about his publication. I would like to invite Dr. Idam Sakti Harahap to lead the seminar. So the time is yours.
Bismillahirrahmanirrahim. Assalamualaikum warahmatullahi wabarakatuh. Good morning, ladies and gentlemen. Gentlemen, today we're going to have a very interesting topic of the seminar. It's about exploring the nexus of science and digital technology, contemporary insight into insect systematic and biodiversity. As we know, biodiversity conservation, including insects biodiversity conservation, is very crucial for sustainability of human life in this world. Insects are abundant, spacious, and involved in many ecosystem functions, and in turn might affect human life quality. This knowledge must be shared to the public. Moreover, in the era of citizen science and development of internet and digital technology. So, in order to stimulate the public to get involved and contribute to the development of knowledge and science, to complete the data about the biodiversity and scientific progress. So the topic discussed in this seminar are very related to those issues. So today we're going to have three speakers. The first one is Dr. Salmah Yaakop, Associate Professor from Universiti of Kebangsaan, Malaysia, as already mentioned by Master of Ceremony. She is as a keynote speaker. And then the second one, Dr. Purnama Hidayat, I should call him Associate Professor from IPB University. And the third one is Dr. Takumasa Kondo, PhD, a senior researcher from Agrosavia, or the Colombian Agricultural Research Corporation in Palmira, Colombia. Dr. Purnama dan Dr. Kondo is as an invited speaker to talk about the issue I mentioned above. So we give 30 minutes for keynote speaker without Q&A, and then for Dr. Purnama and Dr. Kondo is 45 minutes, including Q&A, question and answer. So for audience, if you're going to have questions, please ask the speaker directly or write your question in the chat column. So for saving our time, we invite the keynote speaker, Dr. Salmah Yaakop, to deliver her presentation. Dr. Salmah, the next 30 minutes is yours now.
Okay, thank you. I think I need to share my slide first. Okay, I think the host, please allow me to be the co-host to make me able to share my slide.
Co-host.
Yeah. Okay, you can see my slide now? Okay. Okay, I think I will start it now. Okay. First of all, Assalamualaikum warahmatullahi wabarakatuh. Salam sejahtera and very good morning to all of you. First of all, thank you very much, Mr. MC, Dr. Nulia, and also to the moderator, Dr. Idham Sakti Harahap. And I would like to thank the organizing committee from the Faculty of Agriculture, IPB University, Dr. Ali Nurmansyah, and also Dr. Dewi Satyami and all the organizing committee for this morning. Together with me, two outstanding researcher, okay, Associate Professor Dr. Purnama Hidayat, also from IPB, and also Dr. Takumasa Kondo, Takumasa Kondo, okay, from Agrosavia, Colombia. Okay, to start with, my name is Salmah Yaakop, and I'm from Center for Insect Systematic, Universiti Kebangsaan, Malaysia. Under the Sustainable Crop Management and Development cluster of Sustainable Crop Management and Development, Department of Biological Science and Biotechnology, Faculty of Science and Technology, Universiti Kebangsaan, Malaysia. And this morning, I also represent Entomological Society of Malaysia. Okay, this morning, I will talk about the my title of my speech is the Positions of Insect Systematics, Biodiversity and Conservation in the Citizen Science Era. And as an entomologist, okay, I'm actively conducting research on, of course, on taxonomy, biod, okay, and all the fields, of course, related, involve the evolutionary biology, and involve the several genetic analysis such as the barcode analysis, metabarcode, and genomic analysis. And I think for 30 minutes, for 30 minutes time this morning, I will try my best to relate the insect systematic field, the biodiversity field, and conservation to the and to relate it with the citizen science era. Okay. Ladies and gentlemen, as we know, the insect systematics, biodiversity, and conservation are strongly connected. There are three different fields that are strongly connected. And we know that insect systematics consists of taxonomy and nomenclature. I mean, the process of naming of species, identifying the species and classifying the species. And the insect systematics also is very, very important in making identification of species, and at the same time in estimating the relationship of species. And that's why we need the phylogenetic analysis or molecular clock analysis to visualize the data, to develop the finding, and to see the relationship among species. And this is important because when we talk about insect systematics, it actually is practice, and always get involved with the process of evolutionary biology, precision, and biogeography. And for the biodiversity, actually is a diversification of insect species. And actually there are three types of, this is the common information. Biodiversity consists of three types of biod, is a species biodiversity, genetic biodiversity, and ecological diversity. And this, the biodiversity is very important as one of the parameters to measuring the situation and the current situations of one for the insect species in one ecosystem. And for your information, the insect systematics and diversities may direct to a broader discussion, and one of the aims is in conservation. And we know that the conservation is a careful maintenance and to upkeep the natural resource to prevent the natural resource or the insect species from disappearing or become extinct. Okay, ladies and gentlemen, this is the relation and how we interrelate the systematics, biodiversity, and conservation in the citizen science era. Okay, before that, we have to know that the citizen science era and open science are emerging movements, And actually, they are become significant nowadays and become an important windows or platform for mutually beneficial developments. And however, we need the careful analysis through participation of communities. And that's why the effort of systematics, okay, and diversity to conserve the biological diversity are important. Okay, to make it clear, what is open science? What is citizen science? Okay, so this is information shows the how the citizen science located. Okay, so if you look at here, you can see that citizen science, this is one of the core principles under the open science, together with the other 5 principles. The first one is equity, diversity, inclusion, open data, open access, open educational resources, and open source. Okay, so this is quite important, okay, because we have to understand that we have to know that the conservation, the systematics, biodiversity, and conservation are 3 different fields that really need the open science condition, and because we need to obtain more holistic, more robust findings, and also valuable findings to contribute to more impactful data in order to make the process of management and conservation are very significant and effective. Okay, so when we talk about open science, when we talk about citizen science, okay, and now people are looking at more published papers that are contains more application. I mean, more problem solving. Okay, because rather than the fundamental works. However, in insect systematics work, the fundamental process, the conventional process of identification of insects based on morphological characters are still compulsory. Okay, ladies and gentlemen, this is the situation nowadays. When we talk about the open science era, when we talk about the citizen science era, we need something different to making contribution to systematics toward biological conservation. We need something unique. We need to make it different with the previous one, and to make the conservation and management process more effective and successful. Okay, now science needs more eyes and ears. So what is it actually? So when we talk about the open science and citizen science era, means that we need involvement from many people, from many backgrounds. So we need the collaboration between scientists and people who are interested, concerned, and motivated to contribute within the area. So this is important. Okay, so I try my best to make it simple. Okay, based on these photos, based on this cartoon. Okay, please look at this cartoon, and they are showing the citizens and science here. So means so this is an analogy. Okay, so when we talk about the citizen science, okay, for example here, as an entomologist, okay, as entomologists, we need to obtain more and significant data, very huge data. So how we may obtain this data? For example, when we want to make inventory of species from a secondary reserve forest, for example, okay, and we have to take note about the occurrence of the species. So we need help from the citizen science. Okay, we can get the school children, we can get the politician, okay, we can get celebrities or influencers, and there's many people from other backgrounds, and they may become the hikers, okay, and during the programs of the hiking programs, they will use the specific trails, and during that time they will get some information, and they will note the occurrence and the position where the insects located. So these informations are very valuable to the entomologists, okay, to make sure that they can obtain and collect many data and huge data to come out with the new finding or new insights. Okay, so what is open science and what is a fair data stewardship? Is it something related to without favoritism or discrimination because of fair? Okay, so actually the fair data stewardship is a data management before, during, and end of the project. So means that we need to be fair to all. Okay, so fair to all means that it can be accessed by the researchers, and we can read all the data from the machines, outcome from the machines, and all the data are readable. And we need the guide, someone to guide, to handle the data and metadata, and we need the infrastructure, specific infrastructure in this case, okay, to make it accessible to all the information. And what is a fair data stewardship? What is fair actually? Okay, so the fair is findable, accessible, interoperable, and reusable. And findable means that the data easy to be found in the website. And all the data is not in the website must be put in the website. Okay, and accessible. And we can find it, we can obtain the data free, immediate, and also we can get it online availability of research output, and interoperable, that we can get the data and we may exchange the data. And what's the reusable means that we can obtain the data and reuse it as a secondary data and do the analysis. And at the end of the story, we must share the data. Okay, must share the data to the to many people in the specific website. So this is what I mean by fair data stewardship. Okay. Okay, so now we move to the position. So we try to relate it with the insect systematics, biodiversity, and conservation. So where is the positions of insect systematics, biodiversity, and conservation in the citizen science era? So I think the best way how to discuss, how to elaborate it based on several published papers by researchers on that matter. Okay, and they are focusing on the insect systematics, and they are also focused on the biod, and at the end is the conservation and management of the insects. Okay, for the first one, when we look at here, so the ACIC, and this paper published in 2023, okay, and they have developed tools or the educational frameworks to understand more on the how the insects decline, which may cause the rapid loss of biodiversity and species abundance. And based on this paper, so they come up with the implementation in the programs and etc. to give the public educate the public to mitigate the insects decline. And this is the way how they have documented all the process, all the programs to develop the people abilities and how they connect the social and the insect science in this in that paper, in that published paper. So I think this is one of the interesting papers, and how they come out with the action, the program, the activities, how they can contribute to the open science, and how in the same way how they contribute to the insects conservation. Okay, so this is the first example. Okay, and the second one, when you look at here, so this is the involvement of the citizen science to monitor the establishment and spreads of the biological control agent. So the Arabidae species on the shrubs and on the weed species, from the order Asteraceae. Okay, so in the beginning of the story, okay, the entomologists have documented that species, the biological agent, okay, the Lepidopteran species from India, from Indonesia, from Malaysia, and also from Sri Lanka. Okay, so after the programs of the involvements of the citizen science under the platform of iNaturalist.org, so means that they collected all the information from citizen science, so many peoples, and finally they come up with the expand the distributions of the biological control agent. And now they have mentioned, okay, the distributions of the biological control agent also come from Vietnam, from Thailand, and also from China. So this is the importance how the citizen science, how the citizen science works, okay, through the specific platform of the iNaturalist. Okay, so this is the second one. Okay, so the third one is the review paper. Okay, so the review paper, how the involvements of the volunteers in the scientific research, especially for the entomologists. Okay, so these review papers, they have mentioned about how the citizen science contribute as volunteers to understand the pattern and process of in entomologies, and all the process to understand the pattern is very, very, very inspiring. And in this paper also they have given many examples and how they monitor the pest species, how they monitor the pest species, and how the involvements of farmers, citizen science, how they involve the farmers, immature naturalists, and etc., to give input, to give the specific informations on the distributions, on the occurrence of the species, and many more. Okay, so this is the how the citizen science works. Okay, so the fourth one is a community scientists produce. The title is about the community scientists produce an open data for understanding the insects and climate change. We know the issues of climate change may reduce the populations of the insect species. Okay, and we know that this is also respond to the human cause, global changes, and this is one of the platform. The citizens are using the citizen science. They have come out with the information, the information how to do the mitigation, and how to give the awareness to the many people to overcome these problems. Okay, so this is one of the, I think it's good for conservation, insects conservation. And before they come out with the process of management of conservation, they must be the insect systematics in the beginning, and also measure the richness, the abundance and species under the biodiversity of the species. So this is the involvement of the citizen science. Okay, and the last one, the DNA barcodes and citizen science, involvement of DNA barcodes. So how the DNA barcodes involved here? Okay, how the DNA barcode relate with the citizen science? Okay, and actually when we look at this paper, and this paper is published in 2015, okay, they have mentioned about the one of the species, a ring belonging to the genus Yitima, okay, the Lepidopteran species. And in the beginning of the story, they come out only 7 species based on morphology. And actually they have difficulty to ID the species, okay, based on the ring structures on the Lepidoptera and so on, okay. And after they obtain from the specimens from the citizen science and many from the many resources, they have examined 169 individuals, okay, based on genitalia, based on details on morphology and etc. And they also implement they obtain the DNA barcode information from all the individuals, okay, through through the citizen science programs, and this is under the butterfly project, school butterfly project, and Peninsula Malaysia butterfly count. So this is the platform. So after that, at the end of the story, they come up with several new 3 species, okay, and because they have upgrade the subspecies level to the species level, another 3 species. So the total number now is 10 species. So this is the information that very clear, okay, to how the citizen science and how the DNA barcode using their digital technologies to obtain this information. Okay, and digital is a tool. Digital is the tool of for open science and research. And this is, I think this is one of the interesting part, okay, because when we talk about digital as a tool, and we have to make sure that all the information obtained from using the digital tools, accessible to the researchers, okay, and accessible to the digital science, to the citizen science, I'm sorry, okay. So it means that we need the devices, we need system, okay, and to obtain all the data, And in this and at this situation, okay, we need the machinery programs to come out with the interesting findings, to come out with the significant findings, to help the citizen science, to help the entomologists, to make sure that the programs of insect systematics, the field of systematic, the field of biology, and the field of conservations are effective and very, very valuable and significant. Okay, so please look at this. Okay, so this is the smartphones. So most of the time, every people nowadays have even not looking at the ages, not looking at the difference in level of education, not looking at the economic status. Every one of people have their own smartphone. Okay, so this is the way how they can contribute to the systematics biodiversity and conservation, okay, by making collection of data, okay. So it means that they can taking photos for documentation, they can obtain the coordinate where the occurrence of to record the occurrence of the species, okay, and actually will involve a million numbers of people towards to collaborate toward the same research goal. Okay, so this is how the digital era, how the digital tools contribute to the three different fields. Okay, so this is one of the best papers. Okay, I can see so far. So this is the review papers, and actually they have collected all the several data sets using the digital tools. Okay, so the title is Emerging Technologies Revolutionize Insect Ecology and Monitoring. And if you look at here, they have obtained the data from the computer vision, acoustic monitoring. They did computer vision, acoustic monitoring, radar, and molecular methods. So every single part will contribute to the systematics biodiversity and conservation works. Okay, and if you look at details, every single one for the computer vision, they will get the data from the machine learning technologies. Okay, they can get the abundance information on the abundance of species, interaction of the species at the same times. Okay, and also based on the acoustic monitoring, they also can get the occurrence of the species based on the acoustic information. Okay, so they can record the information using the machine learning technologies, and also based on the radar. Okay, so they can get the biomass information, the body size, body distribution, and movement of the species at the same time. And the last one, I think is very familiar with me, is the molecular method. Okay, so how the DNA barcode and how the meta barcode involve in the systematics biodiversity and conservation works. Okay, so this is because they obtain a huge data. What we can say is the big data. Okay, through the specific platform which is available, accessible to all people. And based on this information, we can get occurrence and we can understand the interactions of species. If we go more details on the discussion on the specific part. Okay. Okay, so this is one of the examples on the I think the applications of the digital tools. Okay, so I think and the last one, the last part is the NCBI board system as the platform for the open science. And I think we have, I have me and my colleague and all my postgraduate students working on this. And we have obtained many data from our research based on DNA barcode and based on meta barcode. And this is one of the what we can say as the digital tools and it's IT, the information technologies. So we know that what is the barcode, we know what is the meta barcodes, and I think there are I think better for me to show several papers, published papers, and what's the current situations and how we get involved with the digital eras. Okay, so actually we have obtained several DNA barcode informations from many species, which is significant for the conservation and management. And we also looking at the species have shows very high variations, and we implement the DNA barcode analysis to the immature stage of the insects to make a quick access, to make quick identification of species. And this is the applications, how we relate the insect systematics with the digital, with the applications of digital tools. And this is also the meta barcode, how we get the informations, the diet of the species, okay, how we get the informations on the microbiome of the species, and all the informations are, of course, they are connected. And also direct to the conservation management, biod and systematics. And actually, there are several papers, books published by me and my friends several years ago. Okay, and I think now we have make it a different change to make it we are relevant to the digital science era, to make it relevant to the open science era. Okay, we are trying to make it every single one is a EISSN. Okay, make it electronic, make it every things can be accessed by many peoples out there. Okay, so this is one of the efforts. Okay, and this is the published, it's a journal published by department, Center for Research Systematics under the Department of Biological Science and Technology, UKM, and we make it open access. So I mean, many people, all the entomologists from Malaysia, from Indonesia, from all the countries may access the journal without limit, and we no need to pay any single cents to access that information. Okay, the last slide from me as a conclusion, to make ourselves as an entomologist relevant to the citizen science era, we need networking and collaboration. Okay, to make sure that we are visible to many people, okay, through our publication, and every single one must be put it in the specific website, and to make it online, accessible to all the people that may get more, may produce more other significance and effective and more valuable findings in the near future. Okay, and I think that's all from me. And thank you very much for the to giving me opportunity for this keynote speech. And I think that's all from me. Thank you very much. Yeah.
Thank you, Dr. Salmah Yaakop. You have 30 minutes sharp. So what we get from the presentation of Dr. Salmah Yaakop is about conservation. Conservation of insect diversity is very crucial. So in the era of internet and digital technology, and also the citizen science, the scientists can ask the community to participate in providing data about the presence of insects in their area. But of course, the community must have access to sources and knowledge about the entomology, including the insect systematics. But I'm sorry for Dr. Salmah because I forget to read your CV. So we can see the CV of Dr. Salmah here. So the expertise is in biological science and biotechnology. And then the qualification or education is BS in the University of Kebangsaan, Malaysia, graduated in 2000. And then MSc from University of Kebangsaan, Malaysia. And then University of Van Amsterdam, 2000 and 2001.
The Groningen. Oh, okay.
It's a program, I think. Yeah.
Okay, what's the program?
Yeah, and then the PhD from State University of Groningen in Netherlands in 2011 in operational biology. So that's from our keynote speech, Dr. Salmah Yaakop. And the next we invited Dr. Purnama Hidayat. Your time is 30 minutes for presentation and then 15 minutes for question and answer. The time is yours. Sorry, before Dr. Purnama Hidayat proceed with the presentation, I have to read the CV. So the expertise of Dr. Purnama Hidayat is insect biosystematic and biodiversity, and also pest management. And the qualification of education is BS in IPB University in 1985, MSc and PhD in University of Wisconsin Madison, USA. And his publication is you can see in the screen. You can proceed, Dr. Purnama.
Okay, but you have to stop sharing. Okay, now I can share my slide. Selamat pagi, good morning, Assalamualaikum warahmatullahi wabarakatuh, everyone. My talk today is outlook on insect systematics and biodiversity in the digital technological epoch. Technological epoch means zaman dengan teknologi. So sorry, I use gado-gado sometimes. Yeah. And some of my talk will be overlap with Bu Salmah. So I think it's no problem because we are talking about in the same area. It's about the digital technological epoch in the field of systematic biodiversity and conservation. And this is my outlines. So in the first slide, I will show you about the challenge in the traditional approaches to insect systematic and biodiversity. So we know that a lot of technology we use today actually give fundamental approach for the systematic and biodiversity. And then I will talk about the advancement in digital and molecular technology in insect systematic and biodiversity. And finally, I will close my talk with conclusion and call to action. Okay, what is the challenge in traditional approach to insect systematic and biodiversity? We know that general approach in traditional insect systematic involves using morphological characteristic and other observable traits. And you might know that insect systematic, including insect taxonomy, started from 18th centuries, and you can imagine how the microscope in that time. So a lot of trait, a lot of character probably cannot be seen, right? But while traditional approach have laid the foundation for understanding insect diversity in insect taxonomy, integrating modern molecular method and digital technology will be as a complement and also, I can say, revolutionize the insect systematic and biodiversity studies. So this is have a very significant impact on the study of systematic and biodiversity. I think some sample already mentioned by Dr. Salmah, but I will add some more later on. You can imagine, so we have to know the character one by one, and of course, this is probably in this time, we need new technology to make it more simple and so on. I have 10 about challenge in traditional approach. The first is time-intensive specimen identification. Not so many people interested in this area because it takes a long time. So traditional approach often involve manual examination. You need a microscope, good microscope. You have to dissect the specimen. You have to make a slide preparation and so on. So it can be time consuming. But of course, some taxonomists like to work in this area. They are passionate in this area. The second is reliant on expert taxonomists. Not so many taxonomists. Taxonomists usually have very specific area, like a certain family, certain genera, or certain group in certain area. So very limited. And when that taxonomists die, for example, in aphid, I think the expert now retired, and now we have a problem because not so many taxonomists working on aphid, for example. So that the challenge, one of the challenge international approach. And you can count with your hand how many specialists or taxonomy in your area or in your country, in Malaysia or Indonesia. You can count with your hand. Not many, probably less than 10. And we know that the number of taxonomists mostly in the Northern Hemisphere, like in Europe, in America, and they don't have a lot of insect there compared to in the tropical area like in Malaysia, Indonesia, or in South America. So basically, we have a lot of biodiversity, but lack of taxonomies. The third is difficulty in handling large data set. Now we can produce a lot of data set very fast compared to many years ago. And the traditional approach, it's struggling to handle this because it's good in the time, but now probably we need a new system with handling of big database, automatic analyzing, and so on. So we have to find a new way, a new tool to handle the large data. That's why there's some database in insect I will show you later on that will produce a lot of data set of insect. The fourth is a limited accessibility of specimen. When you use physical specimen, like in museum, it's only can be accessed by someone that visit to that museum. Alone for museum is not easy because you have to transfer specimen, very rare specimen to other countries, and it's a high risk. But hopefully with new technology later on, we can find out, we can share the picture, three-dimensional picture, and so on. Because using AI now, you can make a three-dimensional specimen and so on. So this is growing now, how to digitize the specimen so we can share the specimen not only the person in that museum, but also all scientists, all taxonomists in all over the world. And the fifth is subjectivity in morphological interpretation. As you might know, that some group of taxonomists consider that species belong to the name of A, and that species belong to the name B, or some people say that like cockroach and mites, it's one group, but some people know that's still different group. So it's really a lot of variation in interpretation of morphology, but that's fine. But using new technology, hopefully later on we can be more objective, because everyone will see the very distinct character. So hopefully everyone will not have a lot of variation in interpretation of morphology. Yeah, so far we have problem with subjectivities leading to potential discrepancies in species identification. Okay, using the old system or traditional system, we only use morphology. And we know that genetic information is very important. We can see the evolution and some cryptic species. We also can identify better using a DNA or genetic information, and that not available in traditional approaches. So this is the what you call it, the challenge in traditional approach. We don't have the DNA or genetic information. The 7 is inadequate for cryptic species, as I mentioned earlier. Many species cryptic, very similar morphologically. But if used only the morphology, we cannot identify the species. We need to dissect, check the genitalia, and some people don't do that. And also we can study something else, for example, using behavior or so on. So we need more and more information, characters. So characters not necessarily morphology. This can be genetic. It can be a behavior. It can be the endosymbiont and so on. So host specificity. So the more information we get, the more accurate our identification. Resource intensive of taxonomic revision. We can imagine since 18th century, people collected a lot of insects using character of color, morphology, and at that time very limited of digital or microscopy and so on. So almost half of insect already described now need to be revised. That some taxonomy say that. Basically, revision is a big task in taxonomy. Not necessarily we need new species or identify new species, but the species that already known so far need to be revised because almost half of them probably identified in 18th century, and yeah, microscope at the time very limited, the information very limited, so it's a lot of work basically. So taxonomic revision will involve updating, organizing, classification, resources intensive and time consuming. That's another challenge. The ninth, challenge in ecological and behavioral studies. Traditional method may not capture the nuance of insect behavior and ecology, limiting our understanding and their role in ecosystem. Now, using automatic sensor, we can do that. So we'll talk later in the next few slides. And the last, not the least, of course, there are some more, inability to keep pace with technology. So the pace of technology lately is very, very growing very fast. Computer, a molecular technology, internet, that will change the game of in taxonomy. So we need to keep pace with the technology. The rapid advancement of technology, including molecular, digital, internet, computer, calculation, evolve very fast. So we need to catch up that with our taxonomy or systematic and biodiversity. Well, after we know the challenge of the traditional technology or taxonomy practices, now we are going to talk about advancement in digital and molecular technology that have in insect a systematic biodiversity. So sorry that typo should not put have in there. Okay, so I make at least 5 actually. This can be more, many of them, but I just compress it in 5. First is digital imaging and microscopy. This area evolved very fast. And then DNA barcoding and genomics, insect ID, mobile application, and citizen science. Ibu Salmah already mentioned a lot for this. Automatization in field survey and emerging AI, then quantitative analysis. In digital and microscopy, you can imagine in 18th century when Linnaeus identified many insects using very traditional, very simple microscope. And in the last few decades, we have SEM, scanning atomic microscope. And the picture in front of us is actually, I don't know. Anyone know this? You might tell in the chat. So this is in the head of insect. This is not what you call it? Not eyes. Well, it can be eyes, but I mean it's not a mata majemuk. It's not compound eyes. You might give the information to me in the chat. So, digital microscope and imaging software enhance precision in taxonomic studies, enable remote collaboration. Yeah, my microscope actually can be connected to the internet, and then someone somewhere can together with me, we chat in Zoom, say what is this? This is the character, and so on and so on. So this is the beauty of new technology in combination of a digital microscope and internet. I don't know, hopefully sometime we can work together with UKM, right? Yeah, so to have a class together, identify the certain insect together, and so on. This is very new. Lately, we not only see the outside morphology of insect, but we can see inside using CT, micro CT. So imagine you have a CT scan, right? You might have that. So our brain sometimes being scanned using CT scan, but we can use it in insect. We call it micro CT scan. So we not only see the outside, the morphological outside, but also inside. It's very new in taxonomy and morphology or physiology. You can see this. This is just using SEM, and this is using micro CT scan. This is amazing. This is the head of insect, and this is using micro CT scan. You can see everything inside of that body of insect. So this is, in my opinion, this is a very significant improvement in morphology, physiology, and insect identification, because we can use that character inside of the body of insect to be used as a character. Of course, we also can understand the morphology, physiology, and so on. here. We use SEM in A and B, and in the right side, you can see inside, in the brain, you can see the brain of insect inside of the head of insect using micro CT scan. Yeah, so this is very significant in morphology, physiology, and also in taxonomy. And we never used this in the last century, so because this is just emerging lately. Of course, DNA barcoding, I think Abu Salmah also mentioned. So in insect, we usually use CO1 cytochrome oxidase #1 because there are 3 cytochrome oxidase in the mitochondrial DNA. And this is basically only short sequence, usually about 600 base pair. And you can see in here, so very similar butterfly. When we sequence it, it's different. So we can see the cryptic species can be solved using DNA barcode. Oh yeah, this is the shell, and mitochondrial DNA is basically part of the cell inside here. So basically, this barcode is based on the sequence GATC, basically based on the 4 base of DNA. And it's been done in many insects, and it's worked very well. In the emergence of next generation sequencing, NGS now is also changing the game in insect systematic and biodiversities, because, well, you can sequence many base of DNA in a short time. Okay, of course, you are familiar with gene bank, so we can retrieve information of a DNA sequence or protein in gene bank. You can also upload your sequence, deposit in there. There's an international barcode of life. We call it eyeball. There's a lot of short sequence of DNA for many organisms, including insect. So basically, the barcode of life is not only the sequence of DNA, it's also the barcode, and also the information where the sample came from, who uploaded, and so on. You might check www.eyeball.org. And of course, eDNA is this is emerging information too, because we know we have a good fast sequencer now, so we can use eDNA, collect the DNA from environment, and we can identify what is in there. So basically, we can identify what DNA in that environment. Species special for insect. There is a insect base. This is database contain 16 million sequences, but only for insect. So this web is only for insect. It provides 15 million coding sequence, more than 700 species, and assemble transcriptome of more than 400 insect species, and so on. So this is very good. Hopefully more and more information in insect base. This is a little bit new. It's relatively new, but hopefully it's growing. So we can just directly collect the information of the insect genomic information in the insect base. There's another website It's called Barcode of Life, bold. And you can see in here, the red is already, this is the area where they publish information of the barcoding of DNA. So we can see some in Malaysia, Indonesia, many in North America, Europe, Australia, some in Africa, South America. Of course, there's a public data portal and so on. So you might visit this port system. There's a lot of information directly about the barcode of organism, a barcode of life in the internet. Okay, the third is insect ID, mobile application, and citizen science. Some already mentioned by Bu Salmah. In the term of key of insect, I think there's a revolution in using a multi-access key. There are several software, but the most popular is Lucid. So this is interactive. We usually use PC, but now it's available in phone. It can be downloaded from Google Play and Apple Store. So there are some key of insect using this in shell phone not available. And this is, in my opinion, this is a revolutionary in identification because using only to a binomial key, you have to go to very long step, but using Lucid can be shortened only a few steps. So this is very, very good. Insect ID, AI bug identifier, I think is it's emerging of AI now, I think. So it's a lot of AI being developed, including for insect identification. It's not all of insect, mostly only big insect, but I think this is good because it's more and more programmer now developing this insect ID using AI. So I think this is good. For example, bug identifier, you can download insect ID, you can download in the store, Google Store, and so on. So this is available already. So this is, I took it from Google Play. And of course, iNaturalist is very popular. So the citizen science available in 56 languages. And last year, it's approximately 161 million observation of plant, animal, fungi. Of course, insect is in there. And in the previous day, in last year, 350,000 users were very active. So this is, I don't know if you never tried this, please try. And you can just make picture with your phone cell and then upload it in here. And in few minutes or in 1 day or 2 days, someone will replay, will give information what that species name and so on. So, and also for your taxonomies, if you want to find something new, sometimes this new species of insect that someone upload in that group, and then you can recognize that this is something new because never seen before and so on. So this is very, very useful. Okay, and the fourth is automation in field of surveys and emerging AI. So data collection, storage, transfer, knowledge extraction, and so on. We can use automated technology, acoustic wave recorder. So a lot of equipment can be put in the forest and then we record any kind of sound. In AI, we can recognize if insect, what kind of insect, and so on. If there's a bird, what kind of bird, and so on. So yeah, we combine this automation, and emerging AI. So we can combine that. Chemical recorder, it's available too. Electromagnetic wave sensor network, wireless sensor, computer audition and vision packet, and so on. So, and we can analyze, and we found the trade, abundant, distribution, and interaction. So computer will do that too. Of course, we have to make it, we have to program it, but basically, based on that automation of equipment, we can produce data for the trade, abundant, distribution, and interaction. So unbelievable. This is the sample. So this automated remote insect surveillance at a global scale and the Internet of Things. So we can put many kind of trap, from make-bill trap, funnel trap, mosquito trap, pine trap, and so on, and connect it to the Internet of Things, use satellite, and then from satellite to the phone provider, and give the alert for population and so on. So this is very good. This is automatic insect monitoring using unsupervised near infrared. So using near infrared, they can record flying insect in that area and automatically report it to the modem, and modem will send to cloud, and cloud send to the phone cell. So this is also
Pak Pur, are you still here? I think Pak Pur will have a problem with the internet. Yeah, Pak Pura, we hope that he can rejoin the webinar. We just wait for a moment.
Halo Pak Idam, what happening?
Yeah, okay, yeah.
What happening, Pak Idam? My internet is cut off.
Yeah, welcome back.
Okay, where I were? Yeah. Halo Pak Idam.
Yeah, you can proceed your presentation.
Okay. Where I was it in here?
I think it's one more slide. Internet of Things. Yeah, this one.
Okay. Yeah, it's raining now in Bogor, so the internet is not stable. Okay. Here basically automated remote insect surveillance. We have, so for example, we have several traps with sensors, and then connected to Internet thing, and then put together to the satellite, and then to phone provider, and then we can get alert from the phone. So I think I know in Indonesia several company developing that system to give early warning about their insect population in the area. So basically the sensor is like this, can be used, this is near infrared. So whatever fly in that area will be recorded. And then this this will record and then report to the modem, and to the cloud, and to the computer, to the phone, and so on. Not only using near infrared, some people use laser beam. So laser beam, there's a sensor. So whenever the insect flying crossing that beam area will be recorded by the sensor. And the last one, I think, the advanced quantitative analysis. So many years ago, I don't think that we can analyze the data, big data like this. So using like R statistic and so on, we can collect all data from many group of researchers, like metadata, and then analyze. Basically, this is research in Jambi, and they analyze many thousands of data together using a computer program. I think mostly is R statistic. We can see we can see the shape of our data, not only the standard deviation, but now also the shape of data can be done easily using R statistic. Also for CCA, canonical correspondence analysis, it's very easy because we can write the code, or we just download, because this is open source. So people, after making code for certain analysis, usually upload in the internet, so we can use it and so on. We just need to put the reference who write this code. Well, this comes to my conclusion and call to action. The first, the remarkable advancement in digital and molecular technology have brought a transformative shift in insect systematic and biodiversity studies. This is very sure. And the second, scientists, policymakers, and conservationists must harness these technologies to improve insect conservation and biodiversity preservation. And the last one, this is number 3, collaborative efforts should be encouraged to further develop and refine this tool, making them more accessible and applicable across diverse ecological contexts. I take one quote. The good thing about science is that it is true whether or not you believe it. By Neil deGrasse. Thank you very much.
Thank you, Dr. Purnama. Whatever Dr. Purnama saying, basically, how to simplify the work of taxonomists in identification, prepare data sets using digital technology and exchange information among the taxonomists. I think that's what Dr. Purnama saying in his presentation. And right now, we have 15 minutes question and answer. If the audience have a question, you can ask directly or by raise your hand, and then we can see. Or you put the question in the chat column. Is there any question for the second speaker? Yeah. Yes, the first question is from Nurfianto in Malang. The question is, what is it? Currently, there are many pest control officer forum group in the Telegram, WA and Facebook, but member interaction within the group is lacking, and it's less visible impact on the farmer and pest control program. How to increase interaction in the group and discussion that is formed so that it can have better impact on farmers or fellow pest control officer? Thank you. That's a question for Dr. Purnama. Maybe you can respond to this question.
Okay, I think for the second question by Pak Nurfianto, how to determine the right planting time based on climatological and meteorological analysis and prediction integrated with remote sensing for identification and monitoring bioecology of pests or diseases. Oh, that's very good and hard question to be answered. Yeah, in my opinion, I think I'm talking about certain plant, for example, rice in Pantura. The availability of the water in there is what you call it? It's not any time. So, for example, the water actually come from bendungan, from dam, and usually government or a local government will arrange when the water available in, especially in the dry season. So, in my opinion, first we have to consider when the water available, and the second is about the pests. I think if we can plant in the same time, especially for avoiding the brown plant hopper in Pantura, for example, I think it's better try to plant at the same time, because in Pantura sometimes the certain area already harvesting and then one area just start planting. So of course in that system, the brown plant hopper from the area that being harvested is moved to the new area. That also happened for our rice borer. I think it's also happened like that. So yeah, basically, at my time, in my opinion, it's really depend on the weather or water availability, and the second is also the pest movement or disease. So in this case, actually, for brown plant hopper or for rice borer, stem rice borer, I think it's good idea to organize farmers to plant at the same time, because otherwise, if one area harvesting and then one area start planting, then the pest will move from the harvesting area to the new planting area. That's for Pak Nurfianto. And the second is, what of the important pest control is the right planting time? How to determine the right planting? Okay. Oh, another question is for Dr. Salmah. Okay. Is that right, Pak Idam?
Yeah, you're right. You just answered the second question, but the first question is how to activate the WhatsApp group of POPT so it will impact the farmer in pest control program.
Okay.
You can read on the screen.
Is that the question? Just a second. How to increase traction group in the discussion? Okay. Yeah. Group discussion of WhatsApp. Yeah, in my opinion, I think it's good idea to have a volunteer in that WhatsApp group, not only farmers, not only a POPT, but also the scientists. It can be from research station or from university, but it must be volunteer. So if the discussion need the expertise from the scientists, then our scientists can be involved in the discussion. Probably not so many scientists will spend the time in that kind of group, but I believe there are some scientists will be happy to be involved or to give the information to the farmers. Yeah, Facebook. I don't know, Telegram, Facebook. The problem is actually not all people use Facebook or Telegram, but WhatsApp almost definitely people will use that. That's my answer, Pak Idam.
Yeah, thank you. So I think Nurfianto asking the question for Dr. Salmah too. I don't know if it's have a relevancy. If Dr. Salmah want to respond. I don't know
Okay,
this. I don't
okay.
know the relevance. Yeah, maybe.
Dr.
Yeah,
Idham,
maybe
I also put it my answer in the chat box. Yeah.
Okay.
Okay.
So the answer is already in the chat box. Pak Nurfianto, we can read it. The next question is there any more? Oh, yeah. Oh, from Harlina in Aceh. Question for Dr. Purnama. I have a data on CO1 sequence from endemic butterflies, and I have not published the data. Now, the data also have not been recorded in GenBank. The problem is the data taken in 2016. Is the data still relevant? And how can I get the data into the GenBank? Thank you. Dr. Purnama?
Okay, Pak Idam. Yeah, I will answer the question from Dr. Herlina in Aceh. Yeah, I think if you have sequence from 2016 is still relevant. Why not? So I upload my previous sequence actually before 2010 and still fine. And how to do it? Basically, it's not directly to the gene bank, but using that's database in Japan. You might send WhatsApp to me directly. I don't know if you can do that because it's too long. The step is too long. But yeah, almost all my student work in sequencing, DNA sequencing, now uploaded to the gene bank through the ODGB, that's internet in bank of DNA in Japan. And there's several steps you have to follow in make sure you sequence already clean, and yeah, basically you have to have a clean sequence and then upload it to the database in Japan, and then that directly connected to the GenBank. So later on, if you have my number or you can contact me by my email, you can check in my website in the department, and I will send you the step-by-step how to upload the DNA sequence to the GenBank. Or I can connect it to my student, because my student just lately also uploaded several sequences to GenBank. So my student might help you step-by-step how to upload the sequence. There's another question too, Pak Idam.
For Dr. Salmah, Dr. Purnama, often identification based on DNA barcoding is inaccurate, especially in morphological very similar species or complex species. What does Dr. Salmah, Dr. Purnama respond to this?
Go ahead, Dr. Salmah.
Maybe both of you.
Okay. Thank you very much. Yeah, Dr. Rosfiansyah. Yeah. Okay. I think what you mean by inaccurate is not inaccurate, but actually this is not what you have done based on the morphological ID. It's not similar. Okay, so it's not matched with your morphological ID. Yeah, it may happen because when we deal with the DNA barcode data, so means that because of the availability of the sequences from, for example, from Malaysia or from Indonesia are very limited in the gene bank. So means that the term is not inaccurate. Yeah, maybe it's because of the variations that highly occur in your species, in your specimens. Okay, and that shows the what you call it inaccurate. So if we deal with the DNA barcode data, so means that we can get exactly what we need. Means that the species, the target species that we thought in the beginning of the process. Okay, so the term here is a variation and limitations of barcode sequences deposited in the gene bank from the for example, from Malaysia or from Indonesia. So very limited research on it. So I think that's my opinion, and hopefully this answers your questions. Okay.
If I can add some more. Yeah, it's happened in Bactrocera or in fruit flies. There are some complex species in there. When we use a morphology, not necessarily congruent with molecular data, and yeah, that's happened sometimes, as Dr. Salmah mentioned. Basically, molecular data is not always can solve the problem, because the morphological character that you show can be sometimes only variation, not necessarily that congruent with molecular data. So that's why a long time ago, a lot of people mentioned that molecular or using DNA sequence is always solve the complex species or similar species or critic species. Not always, but in most cases, yes, it's solve or can differentiate the critic species. But in some cases, no, they cannot. And what you have to do, basically, is just another character and other characters. So character is not only morphology, not only molecular, it's many, many characters. It can be endosymbiont, host specificity, that's also character. So basically, the more character you use, the more accurate your identification. That's my answers.
Thank you, Pak Dr. Purnama. Is there any question? If there is no more question, we can proceed with the third speaker. Okay. So the third speaker is Takumasa Kondo, PhD, is expertise in taxonomy and biodiversity, and his education is CBS from Michigan State University, is Lansing, and MSc in Tokyo University of Agriculture, and then PhD in Auburn University, Alabama. So this is his publication, but unfortunately, he cannot join our seminar today. So he just sent a online presentation that will present in momentum. And so, but he say a greeting to all attendee or all participants of this seminar and say sorry, cannot join directly to this webinar. So this is his presentation.
and identification keys, analysis of variation, synthesis of biological information, publication of all the above. Descriptive taxonomy occupies the working time of most taxonomies and proceeds as follows. There are the species in nature. The taxonomies or the scientists go there and collect, then name and describe the species, classify, and do illustrations, illustrate descriptions and keys with associated biological data. Here's myself in Colombia collecting scale insects inside a Cecropia plant. These are ant-loving scales which live inside the plants and are tended by Azteca ants. These Azteca ants are specialized ants that live inside Cecropia plants. So you can see on the picture on the right side a colony of soft scales. These are soft scales that I collected in Tumaco, Colombia, in the Pacific side of Colombia. Here we have a soft scale insect from Indonesia, from the island of Sumatra. On the left side, we have a typical scale insect drawing with the dorsal side of the insect drawn on the left side, and the ventral side is drawn on the right side. So the dorsum on the left side, the venter on the right side. And on the right side the photo, we see an ant that is carrying these soft scale Takahashi lecanim rotundum. This is a very interesting scale insect. When you disturb the nest, the ants grab the scale insects on these sclerotized plates that they have around the margin, and then they carry the insect to a safe place. Let's talk about systematics. Systematics is the scientific study of the kinds and diversity of organisms and the relationships among them. Taxonomy plus estimates of phylogenetic histories of taxa, so taxonomic groups. Taxonomic names and ranks. Species name has two parts, the genus name and the specific species name. For example, the honeybee, Apis mellifera, may be shortened as A. mellifera, but not as mellifera. Genus and species names are always italicized. In the old days, you could also see this underlined. Species, genus, family, order, class, phylum, etc., or different ranks in life's taxonomic hierarchy. A group at any rank is a taxon, plural taxa. Here we can see an example of a phylogenetic tree for insect orders. 4 things that systematists do. Number 1, descriptive taxonomy. Taxonomies describe new taxa. We describe known taxa and make new classifications. 2, estimate phylogenetic relationships of taxa. 3, study biogeography, which is the distributions of taxa and explanations for those distributions. 4, identify taxa and provide means for other people to identify organisms. Note that identification is not the same as classification, as identification is done using a pre-existing classification. Artworks of all kinds are essential to good taxonomy, especially for describing species and illustrating identification guides, including keys. The definition of illustration. Illustration is the artwork that helps make something clear or attractive, showing by example, an item of information that is representative of a type, a visual representation, a picture or diagram, that is used to make some subject more pleasant or easier to understand. Insect taxonomic artwork may show the appearance of the insect more or less as in life. This is called habitus drawings, and photographs, or represent the important recognition features in a two-dimensional drawing. Here we are looking at a soft-scale insect from Colombia. This is an ant-loving scale insect, or also called a Myrmecophilus scale insect. It's a soft-scale Cryptostigma biorbiculus. It's called biorbiculus because he has these orbicular pores on the dorsum, and so we have a drawing on the left side, and on the right side here we have a photograph of two adult females in life. These insects are found in hollow stems and branches of their host plant, tended by tending ants. So how do we illustrate very small soft-body insects like scale insects? Here we have Obaticoccus viscosa. This is a scale insect that lives inside an encasement in the roots of its host. This is in the USA. So here we have several photographs, one of the encasement. B is the insect showing the dorsal side, and on C we see the insect seen from the ventral side. It's very different. The ventral side has a white powdery wax, and the dorsal side is attached to the encasement, so it doesn't have any of these wax, and it's very shiny and red. So these photographs are very useful to show the insects how they look in life. So this is the same scale insect species. It used to be called Oregmopiga viscosa. Now it's Obaticoccus, so it's the same species that we just saw, the photographs. And these are the line drawings. On the left side, we see the adult female. So typical drawing with the left side showing the dorsum, and on the right side, we're showing the ventral side. And then on the right side, we have a drawing of the second instar female. Why are illustrations important? Scale insects may appear as having very few identifiable features. Here we have two species. On the left side, we have Cryptostigma guajua from Peru, and on the right side, we have Cryptostigma longinoi from Costa Rica. They look very similar. So let's look at a drawing of Cryptostigma guajua. So the drawing shows much more details than the photograph of the insect, because we're looking in detail the cuticular features of the insect. Here we see that this species has very well-developed cone-shaped sclerotic stigmatic plates. This is very characteristic of this species. So now we're looking at Cryptostigma longinoi. In the drawing, we can see that the stigmatic plates are very small. And also on the ventral side, we see that it has some sclerotic pores which are absent in Cryptostigma guajua. So once we compare the drawings, we can see that these are very different species. Taxonomists, systematists study, document, and identify the biological diversity of life on Earth. Systematic research is essential to ecological and biogeographic studies and for solving many applied problems, for example, in agriculture, forestry, and medical entomology. Let's talk about the importance of taxonomy in biological control. In the 2000s, a species of black insect of the genus Paratacardina was introduced to Florida, United States, where it caused a big problem, attacking more than 300 species of plants. The species was identified originally as Paratacardina lobata, a synonym of Paratacardina sylvestri. This species is native to India, so researchers in the United States went to India to look for natural enemies of these black insects. And they brought them back to the United States and released them. However, these parasitoids did not work. They were not able to control the species. With Dr. Penny Gallen, we describe a new species, Paratacardina pseudolobata. And then now we know that this species is native to Indonesia. So if you like to do some classical biological control, we should look for natural enemies, parasitoids, in Indonesia, not in India. So here we have illustrations of these marginal duct clusters of three species of closely related species of Paratacardina. On the left, from the left, Paratacardina pseudolobata, on the center, Madihasani, and on the right side, Sylvestri. And if we look at the distance between these marginal duct clusters, not only the distance, but also the number of these microducts, it's different between these three species. So morphologically, they're very similar, but if you look carefully, you can separate them easily. Many of you probably know the cassava mealybug as a very important pest of cassava worldwide, especially in Africa and now in Asia. It is also present in Indonesia. So on the left side, we have Phenacoccus manihoti, the cassava mealybug. And on the right side, we have Phenacoccus hereni. So these species first entered to Africa with no natural enemies. So in order to do classical biological control, they were looking for natural enemies in the New World. But this was not easy because cassava is found from Mexico all the way down to Argentina. And we didn't know where the cassava mealybug was original from. So in Colombia, for example, we have Phenacoccus hereni, which is, when slime mounted, they're very similar, but in life, the Colombian hereni is yellowish, whereas the cassava mealybug is more pinkish. When you slime mount it, the only difference is the number of quincolloquial pores around the mouth parts. These were very similar, and so people were bringing back natural enemies of hereni, but this did not work well with to control Phenacoccus manihoti. It was many years later that Dr. Bellotti from CIAT that found the natural enemies in Paraguay, and they were able to control successfully the cassava mealybug in Africa and now in Asia. So here we have a drawing of Phenacoccus manihoti. When slime mounted, is very similar to its closely related species Phenacoccus hereni. The only difference would be the number of these quincolloquial pores around the mouth parts. Biological diversity or biodiversity refers to the variability among living organisms and the ecological complexes of which they are a part. The most common measure of biodiversity is species richness, or number of species. Unfortunately, some species are becoming extinct before we even describe them. Some scientists claim that, to a rough approximation, 75 to 95% of animal species are insects. Thus, many recent estimates of the world species richness have been based on the assumption that most animals are insects. So here on the left side, we see a speciescape with fly depicting the large proportion of insects when compared to other animals. The same with the pie chart on the right side, where most of the animals are insects. Sampling in tropical rainforests using canopy towers and canopy fogging has increased the magnitude of previous estimates of insect species numbers. There may be 5 to 10 million insects, although a few scientists have estimated as many as 30 million or more species. Canopy towers in rainforests in Brunei, Borneo, with walkaway about 40 meters above the ground. So we can find many insects high up in the trees. This is a photograph that I took many years ago in Chile as an example of canopy fogging technique. In this slide, we can see some insect collecting funnels, another way of collecting insects in the forest. In this photograph, we can see a simple catch of insects after fogging. Here we have another example of what you can catch in a single tropical forest in one day, in one tree. The most species-rich insects orders are the Coleoptera, the beetles, Hymenoptera, ants, bees, and wasps, Diptera, the true flies, Lepidoptera, butterflies and moths, and Hemiptera, the true bugs. Now let's look at feeding group diversity of extant insect species. There are more species of phytophagous insects than predatory or saprophagous insects. About half of all insect species are phytophagous. There are more parasitic than predatory species of insects. So let's look at the scale insects as an example. There are 8,527 described species of scale insects. Scale insects can be found in various parts of their host plants, infesting leaves, branches, fruits, and roots. They include important pests that can weaken or kill plants either by sucking their sap, injecting toxins, transmitting viruses, or excreting honeydew, which becomes a medium for the establishment of sooty molds. Let's talk a little bit about the biodiversity of scale insects. Scale insects can be found anywhere on the plant, on the roots, on the stem, on the tree trunk, on the leaves, on the flowers, on the fruit, essentially anywhere. We can see here various families, soft scales, mealybugs, and the aspidids, for example. Scale insects can be very cryptic. Here we have an armor scale, Pseudonidia trilobitiformis. At a glance, you cannot see the scale insect, but if we look under the stereo microscope and with an insect forceps or pin, we look under the bark, we can find these the adult females being hidden under the bark. Let's talk a little bit about Myrmecophilous scale insects. These are the ant-loving scale insects. Many soft scales and mealybugs have mutualistic associations with scale insects. For example, here in this photograph, we can see Cryptostigma eureki being tethered by Crematogaster ants. Here we have the same species inside the hollow branches of a tree. Here we have an interesting soft scale, Cryptostigma melissophyllum. This is in Brazil, and this is a scale insect that has a mutualistic association with stingless bees. So it is very interesting because there's a moth that comes and drills a hole inside the branch, and then the stingless bees and the scale insects will live inside those hollow branches. So this is a very interesting mutualistic association between soft scale and stingless bees. Here is another example of a mutualistic association between a stingless bee and a soft scale. So here we have on the left side we have a stingless bee, and here we have the soft scale. On the right side we see a drawing showing these soft scale, Cryptostigma cecropia, a species that we just described last year from Ecuador. How fast are we losing biodiversity? Very quickly, especially in tropical rainforests where diversity is high. Estimates vary from 1 to 50 species lost per day, but estimates are unreliable. There's very few data. It is impossible to establish a realistic rate of extinction when we have no idea how many species exist on Earth. There might be from 3 to 30 million species. Let's look at deforestation in Colombia, where I live. So, since 2005 till 2015, about 100,000 hectares were lost. And if we compare to the years 2016 to 2017, in 1 year, there were twice as much hectares lost in only 1 year. Let's look at the causes of deforestation in Colombia. So there's timber extraction, expansion of the agricultural frontier, livestock farming, illegal mining, transport infrastructure, and illicit crops. In Colombia, deforestation is concentrated in the western Amazon, in northern Santander, and the departments of Córdoba and Chocó, areas with great diversity of flora and fauna, where at least 2,697 species, 503 animals, and 2,194 plants have been recorded. Here is a map of Colombia. If you look at the circles, that's where the hotspots of biodiversity loss is concentrated.
In Colombia, there are 262 species of scale insects in 14 families. There's a high endemism with 56 species, 23%, that are only known in Colombia. In the last 20 years, 27 new species have been described, which is approximately 1.4 species per year. Some species have not been collected since they were described, either due to lack of taxonomic studies and/or habitat destruction due to deforestation. In this bar graph, we can see the number of species endemic to Colombia, which were described since 1852, which are known only from Colombia. As you can see, many species have been described since 1990. Here in this bar graph, we can see the percentage of endemic species that are only known from Colombia and organized by family. As you can see, the families with the most species described in Colombia are Coccidae, Diaspididae, and Pseudococcidae. Now, let's look at some endemic species of Colombia. Here in the photos, we see the Colombian ground pearl, Erizococcus colombianus jacobski. This is an important pest of grapevine, blackberry, and avocados, and other plants. Crypticeria multicicatrices is called the Colombian fluted scale. This is a very large size scale insect. You can see the very large ovisac, which is full with eggs, more than 300 eggs in that ovisac. It is a relative of the cottony cushion scale Iseria purchasei, known as a pest of citrus. These scale insect from Colombia also infest citrus and is very prolific. It's also found on mango, very common on palms, and leguminous street trees in many of the larger cities in Colombia. This is an important pest. This or the third instar nymphs of Laurensella colombiana, a pest of avocado trees in Colombia. The adult is more typical of a monoflevate with fewer wax and larger, like a species of Drosicha, for example. Here we have a photo of the Colombian lac insect, also Tacardiella colombiana. This is a pest of guava. It attacks several varieties of guava in Colombia. Here we have an undescribed species of lac insect, also from Colombia. This was found on soursop, Annona muricata. Kermes colombiensis. This soft scale is very common in Colombia. It's very polyphagous, and it's commonly found on citrus trees and guava trees. Here we have a very interesting soft scale insect, which is covered with white, fluffy wax like a mealybug, but this is a soft scale, family Coccidae. This is Bombacoccus aguacati, a pest of avocado trees. Here we have a very large soft scale. This is Hemilecanium guanabana, the sour soap soft scale. This is a very large size scale insect that feeds on the branches of soursop, Annona muricata, and when the populations are high, it can even kill the branches and the tree. This is a very beautiful soft scale, Foldinacanium multiseptosum. This is a Myrmecophilus soft scale, which is found associated with the Steca ants in soursop and other plants. Here we have a species of Pulvinaria, Pulvinaria caballero ramosa from Bogota. It attacks fig trees, native fig trees in the city of Bogota, where it is an urban pest. Cryptinglycia ica. This soft scale is a pest of rosemary, the herbs. It is very characteristic because they produce a glassy wax cover that protects them. Cryptinglycia corpoica is also a pest of rosemary. Both can be found infesting rosemary in Colombia, but you can see the waxy test that covers the insect is very different, although also glassy.
All right, so I think that's the presentation of Dr. Kondo. So what Dr. Kondo tried to say is about his work, what he has been done with superfamily Coccidea, and emphasize that the importance of continuing the inventory of biodiversity in order to monitor the decline of biodiversity or biodiversity loss due to various factors such as deforestation, infrastructure building, etc. We cannot have a question and answer for presentation of Dr. Kondo because he is not joined us directly here in a webinar. So maybe right now is we continue with the certificate, present a certificate to the speaker The first one is the certificate for Dr. Salmah Yaakop. I think maybe you can accept this certificate. And the second one is for Dr. Purnama Hidayat. This is the certificate. Maybe this certificate will send to you by WhatsApp or email, yeah. And then the third one is a certificate to Dr. Takumasa Kondo. Okay, I think that's all the presentation today. And then I should give back this to the Master of Ceremony. Where is the Master of Ceremony? Bu Dewi? Yeah, okay.
Yes. Thank you, Dr. Idam, who has lead as moderator of this webinar. And I would like give a certificate for you as moderator. So please accept the certificate for Dr. Idam Sakti Harahap. Maybe the committee the operator can show the certificate for Pak Idam. Okay.
Yeah, thank you, Bu Dewi.
Yes. Dear all speaker and all participants, thank you for attending this webinar, which organized by Palm Protection Department and Universiti Kebangsaan Malaysia, Entomological Society of Indonesia, Center of Insect Systematics UKM, Entomology Society of Malaysia, and Agrosavia, Colombia. So thank you for you all for the support. And dear all speakers, all attendees, see you in the next series webinar of Plant Protection Department, IPB University. So may I close this webinar officially? Thank you very much.